before we move on to our next topic, I do just want to point out that it looks like, probably to no surprise to our panelists, that 75% of our audience today are most interested in CRISPR-Cas9 in terms of the gene editing method that they're either using or interested in learning about. Okay, I want to move on to reducing off-target effects of genome editing. And Eric, I'd like to talk to you about the following, and then of course move on to Steve. There are several strategies to lower the predicted frequency of off-target events, many of which can be combined to lower them even further. But I'd like to hear from you, what are the strategies to reduce off-target genome editing? Are we limiting unplanned for effects of on-target genome editing? Can you talk us through that a little bit? I think you might be on mute, Eric.
Thank you.
Sure.
I wanted to start this in a similar line of thinking that Steve had mentioned as well. And so I do think the key here is predicting and detecting the off-target effects first and foremost. And I'm happy to go into detail later on how guides or Cas9 enzymes can be modified, et cetera. But I do think good prediction or detection strategies is the first place to focus. And so one thing is in silico strategies to try and predict off-target effects or identify them. Usually there are some good open source technologies that can analyze guide RNA sequences, which of course guide the Cas9 enzyme to the target location in the genome and can predict potential off-target locations. One big challenge for these sort of prediction softwares is the intranuclear environment is very complex, and so a lot of these softwares focus on kind of a 2D environment of your guide RNA and what can it bind to, but you have to consider the epigenetic environment, open and closed DNA. And so as good as these in silico prediction algorithms are, there should also be experimental strategies, of course, to supplement that. Some in silico examples are Cas-OT, Cas-Off-Finder, FlashFry, CRISPFlash. These have different advantages for the speed that they can search and find off-target effects or other things that might be built into how they identify the off-target sites. There are also ones that provide a score for off-targeting, which are CCTOP, CropIT. Deep CRISPR is an algorithm that does consider epigenetic features, looking genome-wide and finding off-target effects. So different tools will be useful for the specific questions that the investigator has. Some experimental strategies then, these can be divided into cell-free strategies, cell-based strategies, and in vivo detection of off-targets. Thinking about cell-free or semi-cell-free extruseq is an interesting one where cells are mixed with Cas9 and guide RNAs and then mechanically lysed before doing whole genome sequencing. So you have sort of cell-based and cell-free components in that strategy. A lot of the cell-based strategies will insert a sequence into the double-stranded break sites. So, for example, GuideSeq will insert an oligonucleotide into that break site, which can then be enriched for deep sequencing. And the advantage is it can find these sites based on where the breaks occur, but sometimes these strategies will only allow for certain repair mechanisms to implement that oligonucleotide. And so there's advantages and disadvantages here as well. There's also direct labeling of the double-stranded breaks in the case of Bless and Bliss, which directly label the double-stranded breaks as well. In vivo detection that I saw has recently come out is called PeakSeq, which was shown to be able to identify breaks and has done a pretty good job of looking at the at chromosomal rearrangements as well. So as Steve was mentioning, some newer techniques can potentially identify breaks and indels, insertions and deletions in the genome, but also are getting better at identifying larger structural changes like translocations or chromosome rearrangements, which is an important direction for the field to continue building on.
Thanks, Eric. Steve, I'd like to offer you the floor too to talk us through the strategies to reduce off-target genome editing. Compared to what Eric shared with us, different from, what are your thoughts?
Absolutely. So a lot of different approaches have been taken to reduce off targets in the field over the years. And so, there's different strategies that revolve around modification to the guide, modification to the enzyme, and we can talk a little bit about that. But it starts really with just screening the gene of interest. So you start with your gene, you identify where every PAM sequence is within that gene, and then you can generate guides to every one of those sequences and screen them and find which ones are the best editors. And then if you're lucky enough to find, a small handful of editors, you can then evaluate using the tools that we talked about to look for off targets and just kind of rank them with respect to the off targets that they generate. Other things that people have done, as we have done, is to modify the guide. Now, of course, we introduced DNA into the backbone. It's not 100% DNA, but it has a fraction of the residues contain DNA. There are other changes that can be made to RNA, as we've learned from the siRNA field, that there are many different modifications to RNA to protect them, especially when they're being used in the in vivo setting to hide them from exon and endonuclease degradation. So that could be implemented to reduce off targets. And then other changes that have been made are to the enzymes themselves. So people have been publishing over the last half a decade or so, modifications to the enzymes to improve on specificity so that they do lose some degree of on-target activity, but they also lose the off-target. And that's a way to kind of improve specificity. So you do, take a little bit of loss on target, capability. And so, you're depending upon the system, you may want very high levels of, knockout. So as we generate ex vivo cell therapies, these are CAR T cell products or CAR NK cell products, you want as high efficiency as you can because maybe you're starting with, a cell preparation, and that's going to be the product. Another approach is to take iPSCs, make the modifications that you want, and clone out a cell that has the changes, and then you don't have to worry about efficiency. But these are some of the different approaches that people have taken to reduce off targets. And so there are many of them, and sometimes they could be combined, maybe modifications to the RNA as well as a modified enzyme to reduce the likelihood of off targets.
Nice. Okay. And Steve, while I have you, I'd also like us to cover, which diseases are suitable targets for genome editing?
I think it's really limitless. If you have the ability to deliver to a particular tissue, organ, or whatever, then you have the possibility of a genome modification there. If it's ex vivo and you're taking cells out, or you're starting with iPSCs and you're going to differentiate them into something, that's different because now you can really manipulate it. But if you're trying to perform something where you're going to inject something, a lipid nanoparticle or some kind of nanoparticle or a virus, and you want it to go where your gene of interest of knocking out is, then you have to have that capability to deliver. And I think that that's been a limitation for the field, as it was for siRNAs for a long time and still remains as such, is the limitation. But I think that really any disease, theoretically. So what's being used now, of course, is the one, CRISPR-based product that's been approved, or CRISPR-based product approach that's been approved is for sickle cell anemia and beta thal. And so that was successful, taking hematopoietic stem cells and making modifications to them. Now you see there are companies that are implementing virus-based methods or lipid nanoparticle-based methods to do direct delivery of CRISPR technologies to different tissues, particularly the liver, which is so far the easiest in terms of the delivery capabilities. But you're beginning to see some modifications being made, not maybe clinically quite yet, where they're modifying nanoparticles to try to deliver them to different cell types like T cells or cardiomyocytes and other things like that. And that could be by liganding, either through an antibody or peptides or other approaches. So there are even ways to manipulate lipids in a lipid nanoparticle that would favor or bias them to go to other tissues besides the liver, even though you're still going to have a first pass effect. So I think it's really, the answer to your question is it is limitless in theory, but at the moment, I think there are strategic points that have been most successful ex vivo using hematopoietic stem cells, iPSC cells, if you can differentiate them into something of interest, such as NK cells or T cells, and then directly to T cells as we do, as well as macrophages. People are using macrophages to make CAR macrophages and as such. So, and then liver targeting, of course, through lipid nanoparticles. So those diseases can include different types of cancers. It can include now the sort of the newer approach of using those same technologies for autoimmune disease. And then, of course, we have the sickle cell, the beta thal, and then all potential diseases where a protein is expressed by the liver, and there are many, too many to really talk about here.